Good evening. My name is Gabriella Farrell, and I am a student project manager at the Clark Forum for Contemporary Issues at Dickinson College. When we held Clark Forum events in person and on campus, we took a moment to acknowledge that the land on which we gathered belonged to Indigenous peoples for centuries before European settlement. Currently, I am inhabiting the ancestral land of the Lenape, the Susquehannock, the Conestoga, the Haudenosaunee, the Shawnee, the Erie, and later the children of the Carlisle Indian Industrial School. I encourage everyone watching this evening's program to take a moment after our presentation to acknowledge the tribes whose traditional land you currently inhabit. On behalf of Dickinson College, the Clark Forum, the Departments of Mathematics and Computer Science, Biology, the Program in Policy Studies, and the Health Studies program, I am pleased to welcome you to tonight's event, at 10 Qs and 10 Questions in Virology. By the end of 2020, the United States had approved 2 novel vaccines for use against coronavirus. Although speedy vaccine development has given many people hope for safety and health in the near future, ultimately, for vaccines to work, they have to be administered, which poses a variety of challenges. Many parts of the country still face low supply of vaccines, while vaccine hesitancy remains a serious threat to our ability to fight the virus. Additionally, new variants of COVID-19 have led to new questions about vaccine effectiveness, distorting many people's initial hope for health and safety in the near future. I am sure many of you can relate when I say I will forever remember 2020 as a year marked by fear and confusion and anxiety and loss, all caused by the global COVID-19 pandemic. All too relatable is also my own family's experience contracting COVID-19 in the final days of 2020. As if living day to day through the pandemic isn't difficult enough, the feeling of contracting the virus, despite doing everything we could to stay safe, can only be described as a profound pain and an intense panic in which I imagined the worst. I am grateful my family and I are healthy again, and frankly, I'm grateful that we are alive. However, the daily uneasiness and angst over this pandemic has yet to fade and probably won't begin to until we gain more control over this pandemic. Vaccination will be a major step in this direction. However, COVID-19 vaccination in the US still faces many hurdles. For this reason, I am grateful to have Paul Duprex to talk to us tonight about vaccines and shed light on how they work, why it is important to be vaccinated, and how concerned we should be about new variants of the virus. Paul Duprex is a molecular virologist, the director of the Center for Vaccine Research, professor of microbiology and molecular genetics, and Jonas Salk Endowed Chair for Vaccine Research at the University of Pittsburgh. Duprex works on human and animal respiratory viruses such as measles, mumps, and SARS-CoV-2, developing novel strategies to increase the thermal stability and deliver vaccines optimally are key elements in his research. Currently, Duprex serves as reviewer for numerous scientific journals, international institutions, and for funding bodies which have supported his program of research. He co-organizes the triannual Negative Strand Virology Conference and has chaired the Virus Division of Microbiology Society. Duprex is also editor-in-chief of the Journal of General Virology and a senior editor of MSphere. He is a member of the American Society for Virology and the Microbiology Society, as well as a fellow of the American Academy of Microbiology. This discussion-led presentation will be moderated by David Kushner, Associate Professor of Biology at Dickinson College. His research has been published in numerous academic journals, including Journal of Virology, Proceedings of the National Academy of Sciences, and Current Topics in Microbiology. There will be a question and answer session immediately following the program, so please type your questions into the live chat next to this YouTube video at any time. We now welcome Paul Duprex and David Kushner to begin their presentation.
Thanks, Gabby. And welcome everyone for coming out this evening to hopefully have Paul. Paul and I are good friends. We've known each other for a long time. So welcome, Paul. And we're going to try and have a discussion and shed some light on things relating to all this chaos that's gone on in the last year or so. Paul, say hi to everyone real quick.
Hi, it's very nice to be here. I'm looking forward to our conversation tonight.
Yeah, great. Thanks, Paul. Okay, so since there's quite a bit to cover, and of course we'll have time to go over more things during the question and answer period at the end, let's just dive right in. Paul, tell us a little bit more about yourself. We got a great introduction from Gabby, but a little more about yourself and your interest in viruses and vaccines.
Yeah, Gabby gave us a great introduction, and actually also a very personal introduction. And I think that that's an important part of this whole pandemic. She told us a little bit about her family and the challenges that they went through. And I think that that's the sort of conversation we want here. We need to remember that this is something which is up close and personal for many people in many parts of the country. I'm at the Centre for Vaccine Research, as we've talked about, and I've called this this talk, this conversation, we will have 10 questions. Why 10 questions? Well, I really was always that kid, Dave, as you know. Whenever I meet you or whenever I meet some of our colleagues, I will get to know you by asking questions. And in school, I was that kid, and I think as an adult, I'm that kid. And I just love questions. I think questions are such an important part of life. It's how we dive deep into each other's lives. It's how we dive deep into science. It's how we get to know one another. And it's how we get to know science. So yeah, I was always that what, who, where, when, why kid. And I think that that's why I show you my Twitter handle here. The Twitter handle is 10 questions. And I think that whenever we're thinking about virology, when we're thinking about other disciplines, biology, mathematics, there really are big questions that you can ask, probably 10 big questions in any discipline. So what I thought we would do tonight is frame our conversation around my Twitter handle, your questions, and we'll get to the bottom of some of the interesting, intriguing questions about vaccines. You can see the Pfizer one here. You can see just some of what we've been involved in here, some of our nanobody studies. And then the other thing that I love and why I'm so pleased to be with you tonight in particular is because I love, and I think it's so important that we do outreach as scientists. I know that you believe in this yourself. We don't have the justification to do what we do and only talk to the people, only question the people who we are in close contact with in our field. Science is not great science if it's not shared science, and science being shared and communicated with the public is really important. So I told you about our nanobody, here it is here. That was a really interesting paper. This was one of our first images of SARS coronavirus. This is the spike protein on the surface of the cells. We're going to talk a lot about the spike protein. And then there were two real fun things this year. You end up in places that you never imagine that you will be in, with people asking you questions who you will never imagine will ask you questions. This is one of the fun events last year that I did a podcast with Trevor Noah. And again, talking about the importance of vaccines, talking about vaccine hesitancy, why we believe in vaccines. And again, here we have a really interesting Esquire, a piece that they put together where they brought together a number of individuals and asked them questions about the pandemic. So for me, questions matter. And the final question, I guess, is you've heard the accent. The question that I'm sure some of you are asking is, where is this guy from? He's certainly not from Pittsburgh. And I just, again, like to introduce myself. I'm proud of the place that I'm from. This is it in one of the rare days of bright sunshine. This is the Giant's Causeway in Northern Ireland, 40,000 basalt columns, hexagonal in shape, that run into the North Atlantic. And of course, there are numbers of different ideas about how this came into being. There's the scientific way, the questions that people asked about basalt and volcanoes. But of course, we would rather rely on the giants way and think about the mythology of Ireland, where we have these giants who are fighting with each other and getting together across the Irish Sea, building these causeways to do battle. So that's a little bit about me, a little bit about the background, where I'm from, what I'm doing, and where I'm from.
Right. So let's talk a little bit about, your interest in viruses. So tell us a little bit about your interest in viruses, and then, so we actually can talk about the coronavirus in a few minutes. Like, what actually is a virus? Just feel simple and basic.
Yeah, this is a good way to start. Again, you start with the general questions so that you have all of the tools in your hand that you could end up having a conversation about viruses in general. So what interests me? Well, what interests me, as you heard in that introduction, is viruses, respiratory viruses. Whenever I think about respiratory viruses, and whenever we think about respiratory viruses, I just love this image here of this lady sneezing. And of course, this is a staged image, but it just shows you what happens whenever all of these droplets are sneezed out. What's important whenever you're thinking about viruses is you need to think about nanometers, not millimeters, not centimeters, not meters, not micrometers or micrometers, but nanometers. And whenever we think about a droplet in this sneeze, if we imagine a spherical 1 millimeter droplet, and that's 1 million micrometers. Now, whenever we want to think about then viruses, what exactly is the virus? Well, if we have this droplet, we think it's really big, and then we think about grains of pollen. Grains of pollen about 50,000 nanometers. So you can see we're dropping down the sizes. This is important because I'm going to show you a little animation of a red blood cell. It's one of the smallest blood cells, one of the smallest cells in your body, and that's about 8,000 nanometers in size. So remember, if we're thinking about this nanometer world, we're getting down into the smallest cells. Bacteria are cells, they're about 3,000 nanometers in size, and it's really only whenever we get below 1,000 nanometers that we end up where viruses inhabit. And the first thing I show you is tobacco smoke, not a virus. Why is that? It shows you why, whenever we're thinking about respiratory infections, why tobacco is so challenging, because it really permeates deep into the lungs, very small molecules. And then viruses sit between 30 and 400 nanometers in size. So that's important that we get a sense of scale in this biological world. Now, if we look at this droplet, this is a really nice animation. Here's our 1 cubic millimeter. So we're zooming in on this 1 cubic millimeter. And remember, I told you about the red blood cell. Here's the relative to scale size of a red blood cell on the corner of that cube. And this is a virion. This is a single structure of a virus. And because viruses are really small, really simple, they build their coats, they build their outside shells made up of protein. They build it out of many repeating units. So what you can see here as we zoom in on this is the beautiful symmetry, and that virion, that virus, is comprised of these 3 proteins repeated around this shell. And if you think about viruses, one of the best ways I like to tell people to consider them is think of bad news in a protein coat. Bad news. Bad news is the genetic material that's wrapped up in this protein coat. These viruses are bad news because they infect and they cause disease in us. So that's a little bit about what viruses are.
Okay, that's good. So basically we just have this nucleic acid and this protein, and it's these little units. So one of those, of course, is the coronavirus that causes COVID-19 that we've been hearing, of course, quite a bit about since the end of 2019. Now, this particular coronavirus, as I think most people are aware, it's a new coronavirus. It's called SARS-CoV-2. Can you tell us a little bit about this new or emerging coronavirus?
Yeah, so this is a conversation then to answer that question, where do viruses come from? Here's the map of the world, and the word that I have in this for us so that we understand where these viruses come from is zoonosis. A large number of the viruses, the emerging viruses, the pathogens that we deal with, are zoonotic in nature. What that means is they come from animals. And these are a large number of viruses and bacteria. We'll not talk about those in detail tonight, but what we want to talk about mostly here is COVID-19. And this is the disease. You quite rightly said there's a difference between a disease. Remember that COVID-19 is the disease. COVID-19 is the disease caused by this pathogen, this bad news in a protein coat. This is SARS coronavirus 2. It's a really interesting virus. We don't need to go into the real detailed molecular biology of the virus, but what's important to remember is that it's an RNA genome. It's actually a big virus. It's quite a large virus to be an RNA virus. compared to many of the other RNA viruses that we work on. And again, the other thing that we really want to think about is here's this structure. You can see this symmetry to the structure. And the key protein that we want to think about is this one here. It's called the spike. So these are the knobbly bits that whenever you see all those animations on television, whenever the virus is moving about, these bits that stick out, these are the proteins. And again, you can see something important about this protein. Do you see the fact that this is a triangle here, a triangle here? Many viruses, because they make proteins, protein complexes, they make trimers and dimers and tetramers. And of course, because this is a triangle shape, you know that the spike is a trimer. This is where the virus binds the cell. This is the receptor binding domain, and how viruses get into cells is they bind receptors on the surface of the cells, and then they enter the cells and liberate the genetic material into the cell.
Right. So we have a scenario where these viruses are, kind of floating around. They're respiratory viruses. People sneeze, or just talk loudly. We've seen the reports about the issues with folks singing in groups and spreading the virus around really easily because it is airborne and moves easily, especially at close distances. This is all why the masking has become so important. If we do have one of these SARS-CoV-2 viruses get into us, what kind of protection do we naturally have, if any?
So that's the next thing that we need to think about. We need to think about what happens whenever a virus goes inside an individual, whenever it causes that disease. So what we have in this slide is just a cartoon of a virus. And again, these sticky out bits just represent the spikes, the spike proteins. And what we then have in green is the virus, and what we have in blue are our real first line of defense in terms of the immune system, the adaptive immune system, and they're called antibodies. So you see this sort of Y-shaped structure here. And antibodies are really important because the antibodies recognize, and essentially if you think about it being a war, they take out the virus. If we have antibodies which are existing to that virus, then before the virus can replicate quickly in us, the antibodies can neutralize the virus and reduce the infection. It doesn't mean that the virus will not infect any of our cells, but it does mean that the virus will not rampantly infect our cells. And remember, that's the difference between somebody who has a little bit of protection because they might have met a similar virus and no protection at all. That's the big challenge with SARS coronavirus 2. It's a brand new virus. We don't have any antibodies, so therefore our immune systems haven't been trained to see this virus. And because our immune systems haven't been trained to see the virus, then we really are highly susceptible to the virus. Now, because it's a brand new virus, then we have to ask the question, how do we study those emerging infectious diseases? We just can't work with it in a normal laboratory. And what I wanted to show you here is a biocontainment laboratory. It's important to remember that whenever we're working with an emerging infectious disease for which we don't have an antibody, we don't have any antiviral therapies, we don't really know a lot about it, we have to keep our workers really, really safe. So what you see here is in our biocontainment laboratory, one of my staff, and you can see that he's got this personal protective respirator on. All of the air that he's breathing comes through a very, very clean filter. And this is how he grows those viruses. Remember, we have to use cells to grow viruses. Viruses, well, outside of the cell are pretty much dead. They don't replicate. They don't generate new viruses until they're inside a cell. So this is basically the types of plates that we grow the viruses in the laboratory. And then again, I wanted to show you this because what is in this plate? It's hard to imagine. This is macroscopic. Now this is the microscopic. And remember, we were talking about the microscopic world of the cells. So all of these are individual cells. All of these blue blobs here in the middle are nuclei. And the red is the scaffolding of the cell, and then all of this green is the SARS coronavirus 2 replicating inside the cells. So again, remember we talked about a red blood cell being about 8,000 nanometers. This cell is probably about 20,000 nanometers. So a red blood cell would sit about this size beside it. So viruses are small, cells are small, and we have to then look at them using these techniques to allow us to see where they are inside the cells. That allows us to culture the cells, culture the coronavirus in cells. We culture it in tissues. And then, of course, if we want to understand disease, it's really important that we culture it in animals because, remember, this is a human virus, but we don't want to study it in people. We want to study it in really good animal models of disease. And again, in our biocontainment facility, we have these animal models that allow us to explore in time what happens whenever the virus is infecting in a laboratory animals.
That's great. Beautiful pictures, Paul. That's on the left. That's, as a molecular and cellular biologist, we love seeing cellular level imagery. It's just
and I think that's what's important. I think that images speak so much. I think that's why it's very hard whenever people don't have a good idea of what a virus is. I think that level of communication of it's not that difficult to imagine it if you just have a sense of scale in the biological world and you understand what you're looking at there, that those are the cells and that's the DNA in blue, and that cells have shape and viruses infect cells, then it's really, it begins to break the concept of what viruses are.
All right. So real quickly, before we get into the SARS vaccine, the SARS-CoV-2, the COVID vaccine specifics, in general, what is a vaccine by definition? And then why have they been so important in public health?
Yeah, so this is a good question, and I think it's really important, just in the same way as when we're asking questions about what's a virus, we should then ask the next question, what's a vaccine? I'm in Pittsburgh, and one of the things that's really important in Pittsburgh, it really runs through the veins of the city, very proud city of its vaccine past. This is Jonas Salk. Jonas Salk developed the first polio virus vaccine. And what he did was he grew a virus just in the same sort of cells that I showed you in that previous slide. He grew lots of it. He grew bottles and bottles and bottles of it. And then he worked out a chemical way to inactivate that virus. And remember what we said about antibodies. Antibodies are generated and they protect us against the disease. So basically, whenever you think about a vaccine is a weakened or it is a killed form of the actual pathogen. So in this instance, Jonas Salk was working on poliovirus. And what he did is he inactivated poliovirus, and then he injected poliovirus, immunized kids with the inactivated poliovirus vaccine. And what happened in those kids' bodies was they were trained to recognize poliovirus that didn't cause a disease. And what does it mean they were trained? Well, their immune system made those antibodies, those blue Y-shaped things we looked at on the previous slide. And those blue Y-shaped things were ready that if those kids met real wild poliovirus in a real swimming pool in Pittsburgh or anywhere in the country, because poliovirus was a real scourge 65 years ago in the States and all across the world. They had antibodies which already recognized poliovirus, and they were able to neutralize it, take it out, stop it replicating, and stop that horrible disease that led to children and young adults being present in these iron lungs. I don't know if you've seen these pictures, and people who had paralytic poliomyelitis. And this was a tremendous achievement. The clinical trials, we'll talk a little bit about clinical trials later. The clinical trials for that poliovirus involved 1.8 million children, massive clinical trials. So that's what a vaccine. A vaccine is something that trains our immune system to be ready for the real virus without causing a disease.
So do you want to take 1 minute to tell us a little bit about, this is great, there are vaccines, and of course we're trying to get some good ones for COVID. We have a few already, but what happens when there isn't a vaccine? So why do we actually need the vaccine?
Well, what happens when there isn't a vaccine is nicely illustrated in this cartoon. Before there was a poliovirus vaccine, and this time last year, before there was a SARS coronavirus vaccine, this is what we looked like. The society looked like all of these blue individuals, healthy people who weren't immunized because there was no vaccine. And then what happened was people traveled, people came into communities, people who weren't immunized, who were sick and contagious, and the virus spread like wildfire, highly transmissible virus. And all of the people in close proximity, that if you can think about this as a super shedding event, this super shedding event led to lots of people getting infected. That happened for poliovirus way back when. And this is exactly what's going on in the United States and elsewhere in the world. In the absence of a vaccine, when no one is immunized, lots of people get the disease and lots of people become sick. Lots of people are contagious. And because we didn't know a lot about COVID-19, a lot of people ended up in hospital. And unfortunately, a lot of people have died of that disease. I mean, the tragedy that we have at the minute is we passed that awful number of 500,000 Americans who have died of a disease that we did not know about January 2020. It's just incredible, so sad. And that is what happens whenever there's no vaccine. That's why vaccines are so important.
Yeah, and they have worked in the past, right?
Yeah, they have worked in the past. This is a good example of how they work. We don't need to go through all of the details of this, but you know that we get all these sorts of different vaccines. I work on measles and work on mumps, and you can see that what happens is in the pre-vaccine era, lots of people die of those diseases. Rubella and smallpox and tetanus. There's a virus, smallpox, that is now completely gone. You see 100%. We completely have no cases of smallpox now because the virus is actually being eradicated by the use of a highly efficacious vaccine. So it's really important to remember that vaccines work. And then the real question is, well, what are we trying to do with SARS vaccine? So I've shown you this before. We've talked about it all being about the spike. Remember, the spike is on the outside of the molecule. The spike is the protein that allows the virus to get into the cells. So what we're trying to do with making vaccines and fighting that disease is we're trying to train the body to make antibodies which recognize the spike, so that those antibodies are primed and ready to go, so that if you meet coronavirus on the bus, or if you've entered a place where someone is asymptomatic and they are not wearing a mask and you unfortunately meet them, that you're ready to fight that infection because you've got those antibodies which are basically ready to go for the infection. So it's really important to understand what's a virus, it's really important to understand what's a vaccine, and it's really important to understand that antibodies in this instance are really important to neutralize that virus, take it out, not completely protect us by sterilizing us, but reduce the level of infection to a state where we don't end up in hospital, we don't end up in a ventilator. So that's the basic premise of vaccination.
Right. So when we're talking about the COVID vaccines, I think most people are probably aware of the term mRNA vaccine. It's certainly been in the news. I mean, it's in the news daily for months now, and probably people are familiar with Moderna and Pfizer as the producers of the very similar to each other mRNA vaccines. But what does this actually mean? And this mRNA vaccine is also kind of a new concept for vaccines, although we've been using it actually a little bit longer than people think. So how are we actually making these vaccines in this case to try and get those antibodies to be present in our bodies to protect us?
Yeah, so that's the next bit of the puzzle that we need to talk about. Remember, we talked about it's all about the spike. So if we basically start at this end of the cartoon here, remember that our main goal is we want to make antibodies. We want to train the body to make antibodies against SARS-2. We can do that in another way, a number of different ways. Central dogma of molecular biology. We don't need to go too deep, but all you need to remember is DNA. We have a genetic material, which is DNA. That DNA is transmitted to RNA, messenger RNA, And that messenger RNA, there's millions and billions and trillions of messenger RNA molecules already in our body. That messenger RNA is what is used to generate the protein. So if we want to make the protein, we can produce the protein, we can inactivate the virus, just the way Salk did, and we can inject protein, spike protein, or we can inject inactivated virus into a person as a vaccine candidate, and that should make antibodies. But what's really interesting about the RNA vaccines is we go one step back. So the Moderna vaccine, the Pfizer vaccine, we don't put in the protein, we put in the RNA. And what we do with the people who are vaccinated is we basically turn them into the factory. We use their cells, because cells are making proteins from RNA all the time. And we just put a large amount of RNA, which encodes the spike protein, into that person. We inject them with the RNA, and the RNA then is taken by the machinery. The protein is made, the immune system sees the protein, and you get really good antibodies against the SARS-2. And this is a fundamentally new way to make vaccines. There's never been an RNA vaccine licensed until the two which have been emergency use authorization licensed for SARS-2. That's a tremendous achievement in such a short period of time. You're right whenever you say that it didn't come out of nowhere. What's very, very important for people to understand is there's a ton of basic research goes into this, how to get the RNA made, how to wrap it up in a safe way to get it into your body, how to keep it protected so that lots of proteins get made. So all of that work was done beforehand. And then again, another way of making vaccines, and this hasn't been as successful as the RNA vaccines, is DNA vaccines. So instead of putting in RNA, you put in DNA, and remember, DNA goes to RNA, RNA goes to protein. So those DNA vaccines would do the same job, make proteins. That hasn't been as successful for SARS-2. We don't have a licensed DNA vaccine, but again, people are working on those different modalities to generate antibodies. So remember, what we're trying to do is we can make vaccines in ways that Salk could only have dreamed about whenever he was inactivating poliovirus. DNA goes to RNA, goes to protein, and the job of all of those injections is to generate the protein in the body and to then let the body produce antibodies against that spike protein. So we have lots of different ways to make it, but we don't only have DNA vaccines, RNA vaccines, and protein vaccines. We also have the ability to harness viruses themselves. So we've talked about Moderna and Pfizer, but we haven't talked about the Oxford vaccine or the J&J vaccine, or we haven't talked about the Sputnik vaccine. We talked about that a little bit. You've heard that in the news, the Russian version. These are adenovirus-based vaccines. And what happens here is we take these adenoviruses, which cause the common cold, we put the DNA, which encodes the spike protein, inside these adenoviruses, and then we inject large amounts of adenoviruses into people and use the adenovirus as a DNA carrying virus to deliver a vaccine. And then there's other different ways. Vesicular stomatitis virus can be used to make vaccines. That was the one used to produce the Ebola vaccine, an interesting recombinant virus. And then we work in my laboratory on measles virus to try and take measles, a wonderfully safe vaccine, and use that to produce the SARS-2 spike proteins as well. So we end up with lots of different approaches. Moderna, BioNTech, Inovio does DNA vaccines, AstraZeneca does the adenovirus. We're working with Serum Institute of India. You've heard about Johnson & Johnson. IAVI and Merck are working with the VSV and the measles platform as well. So there's a real renaissance in vaccine development, lots and lots of different ways to make vaccines against this pandemic virus. So that's where we sort of sit.
This is always really interesting to sort of consider and think about. And I still think that most folks keep hearing about how fast this happened. I mean, it did happen in a year, actually a little less than a year, basically 10 months or so to get the Moderna and Pfizer vaccines up and running, which is really, really impressive. But there was, as you mentioned, Paul, a lot of work that was done beforehand. So we were in a position to take advantage of that knowledge and use it in the vaccine development. So normally, or even last year, you kind of stage the process of vaccine development and test it out and make sure it's really, safety is really the first thing we're trying to check, right? So can you talk a little bit about that?
Yeah, so one of the things which is very important is these, as you say, these vaccines don't come from nowhere. A lot of work has gone into them. We understand a lot of the biology behind what we're doing with these rational ways to make vaccines. The next question that we have to think about is how do we test them? We talked about the development. So I've talked a lot about these in vitro studies in the laboratory, making these recombinant viruses or RNAs that you're going to deliver, or DNAs. We haven't talked a lot about the in vivo preclinical studies in animals, but that's also really important before you license a vaccine. Before you go into people, you really want to understand that the vaccines work in a good animal model. And I talked a little bit about that at the start, but we haven't focused on that in terms of the development and preclinical stages. What happens next in pre-licensure is the really expensive, really slow parts of vaccine development. That's the phase 1, phase 2, phase 3 trials before we get to licensure. These are really expensive. And as you have said, the most important thing to do in the first instance is determine whether or not they're safe. Now, how did we do it so fast? Well, we compressed the whole timeline, where we put a lot of money and a lot of effort into it so that people were planning phase 3 trials before the vaccines had been tested fully in preclinical studies in animals, so that you would never spend money here if you didn't know that this had worked. But because this was so urgent, we were able to start this process here and be ready, even though we didn't know if the preclinical studies would work. So that's how it was compressed. But remember what's really important: no corners can be cut. Vaccines are way, way too important to the public to cut any corners, to do anything rushed. There's a big difference between rushing something, the idea that you're cutting corners, and doing something fast. And we really have done this very fast. So phase 1 is important, where we have tens of people. We check in healthy adults whether or not that is safe. Second stage of the process, we check in hundreds of people. Is it safe, and does it generate antibodies? That's phase 2. And then thousands of people, we ask the next question: is it safe? Does it make antibodies? And does it work? And the term that we use for does it work is efficacy. Is it efficacious? And then what we get to is we license the vaccine. The licenses at the minute are for emergency use. And even after the vaccine is licensed, we carry out post-licensure checking to determine that there are no surprises whenever those vaccines are given, not to just thousands of people, but hundreds of thousands of people. And remember, now in the UK, we have 18 million people in the United Kingdom are already vaccinated. So we learn a lot during the process of developing the vaccines. Efficacy really matters. This is just to talk about one of them. This is the Pfizer and Moderna one. And what we were able to know at the end of last year was that that whole pipeline, that whole process, it was over 90% effective. And that is just incredible for a new vaccine modality, that we stop people dying, we stop people ending up in hospital because of the delivery of these RNA vaccines. So that's what efficacy means. And now we can then think about our little cartoon. In the early stages, there's not enough vaccines at the minute for us. So the question here is, what happens whenever a vaccine is developed? So you can see that there's lots of people here who are not immunized, but they're healthy. We have these 2 red guys here who are infectious. But you see here, we have some of the population getting immunized. And whenever we get some of the population immunized, not everyone gets the disease, but because there's only small numbers of people, they're still getting immunized whenever a vaccine is developed in the early stages. The virus still spreads, and that's what's happening at the moment. But then what happens whenever we roll out the vaccine and more and more and more yellow people are over on this side, you see here? So we have lots of yellow people, lots of people who are immunized and healthy. We still have a number of people bringing in the disease. These guys here in red again are the infected ones. But you see here and here, the likelihood of these people getting infected is very small. Most of the population gets immunized, and only one of the blue guys becomes infected. We still have 3 here instead of 4. And we have 1 person infected. So what we do is, because we've vaccinated a large number of people, we really reduce the amount of virus which is spreading in the community. And that's why this little cartoon, whenever we spread the virus, is really useful. This is a cartoon to show you from 0 through 25, 50%, 70% vaccinated, 90% vaccinated, and 95% vaccinated. So you can see if we have 1 person with no vaccine, it spreads like wildfire in the absence of a vaccine. Then whenever we vaccinate a quarter of the people, you can see some people are fine, but the virus still is able to spread because there are enough susceptibles. And as we increase the vaccination, we stop the virus spreading, so that whenever we get up to 90% of the people getting infected, this is for measles, we really break those events. So instead of having this massive cloud of people here, we have a significant number of people protected from the disease. Now, the other thing that's really important to remember whenever we're thinking about vaccines is when we have a good vaccine, and most people can get immunized, those people are healthy individuals, they don't have comorbidities, the vaccine is not contraindicated. Contraindicated just means you can't give that person a vaccine. So what we have here is say for example, a child who has an immune deficiency. And that child who has an immune deficiency is part of the community, part of that population. They just can't get immunized. But if we think of vaccines as being a social contract and most of the population gets immunized, even whenever we have some individuals in the population who are infected, they will transmit to only 1 other person here. But because the majority of people are vaccinated, and because the chains of transmission from person to person to person to person to person are broken, this individual here, this immune suppressed child, is protected whenever they can't take the vaccine. And again, that's a really important thing to consider whenever we're thinking about vaccines. It's not just about vaccinating yourself and protecting yourself. It's doing the right thing for all of society, because there are some people in society who can't be vaccinated. That's an important concept to get as well.
Great, Paul. A lot of information those last few minutes. It's all really, really important to understand and appreciate the effort that's gone into ensuring safety and doing the multiple stage clinical trials in order to get people to be willing to get vaccinated. And so, we're starting to move forward, of course, in the US, but we still have a long way to go. And of course, then there's one little thing that's been cropping up that's been in the news that people are concerned about, which is the reports and the fact that this is happening now in the US. We have, coronaviruses and SARS-CoV-2 being RNA viruses. Those RNA viruses tend to mutate. And so what can happen is we get these things called viral variants. So tell us briefly a little bit about what is a viral variant, and should we be concerned about this? Or maybe is it being a little bit overly hyped in the news as a concern? Like, what's the real concern here, if there is any?
Yeah, so this is, again, us getting to the end of our question, getting us ready for our question and answer. And we can go through this in greater detail if people are interested in the question and answer. But just to bring these variants to the forefront of our conversation, you have to remember that viruses evolve. And if you're a virologist like you and me, what we see is evolution up close and personal. We see it in front of us every single time we grow a virus. And whenever you're thinking about evolution of a virus, it's really a game of cat and mouse. The mouse runs ahead and the cat tries to catch up. The mouse is the virus, the cat is the immune system. And what you don't want to happen is the immune system to get too far behind, because the mouse has changed, it's got into a different position, it's got further away and the immune system can't see it. Or even better, the mouse has evolved to run 10 times faster. And that's the best way to think about the variants. The variants change, they look different to the immune system. And because the viruses evolve, the variants might be harder for the existing virus vaccines to neutralize these variants. That's why good biology, good virology, and keeping a very, very close eye on where the virus is, where these variants are coming from, really matters, because we might need to then design next generation COVID vaccines to be able to protect against the variants. So if you think about that, we have to think about second and third generation vaccines. What's the current state of play? Well, it's more than just Moderna, more than just Pfizer. We need to have other ways of thinking about new Pfizer vaccines, new Moderna vaccines, so that we're able to keep on top of these new variants. So that's why the pipeline, whenever we think about developing vaccines in this development and preclinical phase, the pipeline has to be full of novel, different ways, because what I often say is first may not be best. What does that mean? It might be very efficacious, but one of the big challenges is if you have to keep the vaccine at very, very cold temperatures, that's a hard vaccine to roll out to all of the world. And the difficulty with rolling out, not being able to roll out a vaccine to all of the world is, remember what we talked about at the start, these zoonotic viruses, a case somewhere in the world because of the way we travel, ordinarily is a case anywhere in the world. And that's why developing vaccines for all of the world really matters.
Right, one more quick question before we sum up so we can get to the Q&A period of tonight's event. A lot of people have been fortunate and have received their 2 doses of vaccine, which is wonderful that people have reached that point. And people, of course, are asking, so can I throw out my mask and can I stop social distancing? And can you briefly tell us a little bit about, can you throw away your mask yet? And what about social distancing?
Yeah, so even though we have come a long, long way pretty quickly, we also know that the vaccines don't sterilely protect us. Okay, that's a very important concept to get. The vaccines dampen the disease, but we can still replicate the virus. So therefore, it's a good idea to keep using those environmental ways to block the infection, staying 6 feet away from people, wearing the mask. It's biology is not black and white, right? It's not binary. There's a lot of grayness in biology, and transmission is very hard to understand sometimes. But what we know is, just think back to that sneeze, that lady, that picture I showed you at the beginning. If that lady had a mask on, would those droplets be going as far? Absolutely not. Would there be some droplets going through the mask around the sides? If she did that amount of sneeze, yeah, probably, but it would reduce the likelihood of transmission. So that's the way we need to think of it. We need to stop thinking about this yes, no. That's why it's hard for people to think about 90% efficacy, right? What does that actually mean? whenever, a drug either works or it doesn't. That's the way people typically think. Vaccines are rather different in how they are considered. So yeah, just to summarize, I guess, we talked a lot about vaccines. We showed Salk and all of that knowledge which has been built over all of those years, from poliovirus 65 years ago to COVID in the here and now. We just are living in as I said already, a renaissance of vaccine development. So much creativity, so many new ideas, so many different ways to fight these old threats. It's really quite interesting to be living at this particular stage, to be vaccinated by something which has never been used in people before. I think it's an exciting time to be doing science. But again, back to what Gabby told us at the start, this is also a tragic time to be living. This is why doing the right thing for all of the society. I know that people have questions, people are hesitant about vaccines, but it is really important for us to champion vaccines and to answer those questions whenever people are hesitant. I think it's really good. This is all about questions. We've gone through lots of questions about vaccines. It's really good to ask questions about what's in a vaccine. What is an RNA vaccine? Why is my arm sore? Does it give me the disease? All those things are really important to ask. And that's why dialogue and things that you're doing tonight are really useful because they educate us, they allow us to communicate and again, just to champion that vaccines work. They've really done so much for public health. So there you go. I think we got through it. We've got to questions and we should probably jump and hear how we've confused people or not confused people and what people need to know and what we need to amplify. So over to you for some questions, Gabby.
Yes, well, thank you both so much for that conversation. I had so many of my answers, so many of my questions answered in that. So thank you. So as promised, I'm going to start with some questions that are coming up in our YouTube live chat. The first one is, it asked, Paul, do you think, as does Ralph Barrick does, that through a combination of natural infection and vaccine-induced immunity, that SARS-2 will eventually become another common cold virus like OC43?
So that's the really interesting question at the minute. Sorry, I'm just trying to get to see you. one little second that I can actually see. Yeah, I think that what we are looking at is a virus which is essentially going to be with us for quite some time. We know that we live with coronaviruses. There are other human coronaviruses that are not as virulent. Virulent is basically disease causing as SARS-2. And remember that they have co-evolved with us over quite some time. Because we are living in a world where we can track viruses now, we know a lot more about what viruses are, where they come from, sequencing their genetic material, all of these things. We haven't been able to look back in history whenever those historical coronaviruses that are with us at the moment, we didn't see what happened when they jumped out of animals into people. But what we see is the evidence that over time they basically became endemic pathogens in human populations. And I think that's a very reasonable assumption to make that SARS coronavirus 2 will be something that we will live with for quite some time. The real question is, will we have to vaccinate again and again and again, and how often we will vaccinate? And one of the things we didn't really touch on, because we don't know, is how long the immunity lasts whenever you are vaccinated. All of those things we learn at the moment.
Awesome, thank you. And this next question is kind of related to that, about, what we should expect for the future. I'm going to try to summarize it because there's a lot of biology stuff that me as an international studies student, I don't completely understand, but it's basically asking, it's a biochem and molecular biology student here at Dickinson who's interested in pharmaceuticals. And they're asking if you think that it's possible to create a drug that maybe inhibits, acts as an enzyme inhibitor to the ACE2 receptor, and therefore prohibits the COVID-19 virus from entering. And then they ask, or do you think that the vaccine, once it's widely distributed, if that would mean there isn't a need for this possible therapy?
Yeah, I'm a great believer in multiple approaches. I think that one of the things that we have to be very careful about is just assuming that everything is finished whenever we've got a couple of vaccines. One of the most important things in pharmaceutical world, I used to work for Johnson & Johnson, and they have backups of backups of backups. We talked a little bit about the pipeline. You can't assume that everything is going to work well. And again, we don't know that the vaccines that we have today are going to be working next year. That's why I think antiviral drugs and other interventions, like for example, antibodies, we didn't talk about. We talked about antibodies which were generated in your body after a vaccination, but what we didn't talk about tonight were antibodies that can be generated in the laboratory and injected into you. That's Regeneron. If you're hearing about companies that make those sorts of things, that's a company that makes antibodies that can be injected. And I think that antivirals are a really great idea. What's really interesting is antivirals have been very hard to make. So we have had phenomenal successes with some antivirals and failures with vaccines. So look at HIV. HIV, we've known about that disease, that virus for many years, and we do not have a vaccine, but we have really good antivirals, combinatorial therapies that allow HIV to be something that people can live with, as opposed to people that died with it many years back. Hepatitis C is another great example of where we have antiviral drugs, which really have taken that and revolutionized how people can live, survive, and have that virus gone from them. So it's really interesting, and that's why it's important to understand that the viruses are all different. Different things for different viruses. Antivirals are hard; they're not impossible. Vaccines are good, but for some pathogens they're really difficult. HIV is an example. So I think lots of different ways to deal with the same problem. I think humans are creative. I think it's just one of the attributes of a human. Humanity is incredibly creative and innovative, and that's where I really love science, because it gives me an opportunity to be creative in my world. And I just think that basic science, people exploring industrial science, people exploring new ways to treat old infections, really important.
Real quickly, I'll just add, one of the benefits of the vaccine is that it serves to, for most people, because it's not 100% effective, but for most people, it's to prevent the infection, prevent the virus from really getting into you and making more of itself and spreading. So if you can do that, you don't have to rely on the antiviral therapy, which you would be taking if you really did get a large viral load and maybe got some disease. But it's great to have them if you can develop them as a backup.
Yeah.
Okay, cool. That makes sense. So I have a question. My dad, he actually texted me. I think he's too shy to write it in the chat. But so, as I said in the introduction, my family, we contracted COVID in December. And so now that we have it, it's a little bit confusing. What people who have had COVID, what does that mean for vaccination? Are we getting the same amount of vaccination? Is it going to be any different? Are we going to be put at the bottom of the list? Like, what does that mean?
It's actually interesting. I had a conversation yesterday with someone about conversations that are being started about only vaccinating. Now, remember how we did those clinical trials? The clinical trials were not done with single shot mRNA vaccines. It was double shot. But one of the conversations that people are thinking of doing clinical trials is for folks who have had the disease, vaccinating them with a single shot, because remember, your body has already been trained to make some antibodies. And if there was, for example, a way of increasing your antibodies that had been made, you would be taking advantage of the fact that you already had some, boosting them with a single shot, and you might get to where I am because I haven't had the disease. You and I might reach the same levels. Your dad and I might reach the same levels. You with 1 shot and me with 2. So that's a conversation that people are having, and there's going to be no reason that I know of at the minute that folks who have had the disease will be barred from getting the shot. And again, remember, we have new variants coming along. If we have to get new vaccines out, then there would be absolutely no reason why folks like your dad wouldn't be vaccinated against those new variants. So that's where it's hard. It's sort of work in progress, but nevertheless, it's something that we do think about.
Thank you. So another question we have is, someone wants to know what type of animals are used to perform the preclinical trials.
So in the laboratory, you use a wide range of lab animals. One of the best ways to detect if antibodies are raised can be in murine models. Murine models, well understood. We understand a lot about their genetics. We understand a lot about their immune system, and we understand a lot about how to give vaccines and test vaccines in them. So those are really well used. One of the big surprises of the coronavirus, and again, this is why we need to be creative, is the hamster model has been really very, very useful. Hamsters, for some reason, have a very, very nice way of being infected. Nice, you understand what I mean by nice. They're naturally infectable. They recapitulate the disease, and you can take an antiviral or a vaccine, give it to the hamster, and determine that the hamster is protected. And then we use other models. Influenza virus gets tested in ferrets quite often. Ferrets are a really good model for influenza. Ferrets sneeze. Remember what we're talking about, these respiratory viruses, they have a much more similar system, respiratory system to people. So therefore ferrets can be really useful for influenza. SARS has been tested in ferrets as well. And then there's a wide range of other animals that get used in the laboratory. As you increase the type of animal that you use, the larger animals you use can be quite expensive. For example, people tested pig models for SARS as well. And then many of the vaccines ultimately have to be trialed in non-human primates. Non-human primates are used very, very judiciously. It's really important to be, I'm an animal lover, you know, I study disease, but I just love animals. So I think that you have to really have, whatever you're using, whether it be a mouse or a non-human primate, you have to have the highest ethical standards for animal research. And again, we do that because we have to test the vaccine modalities, the drugs in animals before we just go straight into people, because we just cannot get to the point of something going wrong in people, and all of that preclinical studies really matters. So there's no real answer to that question. There's a lot of different species used in the laboratory. We use the best species for the best question. Again, back to the questions, best species for the best question to answer the question well, so that we reduce the number of animals that need to be used. It's a very important part of biomedical research, reducing the numbers of animals. We don't want to waste sentient beings, for sure.
Awesome, thank you. That's really interesting. So I think we have time for one more question. This person's asking, what type of vaccine is the Sputnik vaccine? And do you know anything about how well it works?
I don't know a lot about how well it works. I understand that it is an adenovirus vaccine. So if you remember back to that slide where we had the pictures of DNA goes to RNA goes to protein, and then the adenovirus is the one which is this hexagonal shape with these long fibers that stick out of it. So the adenovirus is, the Sputnik one is very similar to the Johnson & Johnson one, the Oxford one, and can't remember who has another adenovirus vector. But yeah, that's it's an adenovirus.
Thank you. Well, like you said earlier, I wish we had more time to keep talking about this. It's so interesting, and I feel like I said, like before, I wouldn't have really cared so much, but now it's so important, and I think I learned so much, and I'm sure everyone else in the audience did as well. So, thank you both so much for your time tonight, and for coming to talk to us about this. That's all we have for tonight. So, again, thank you to our audience members and for everyone who submitted questions. And I hope everyone has a great rest of your night.
Thank you very much for the invitation. I really enjoyed it.
Thank you.
Thank you, everyone.